Species have been evolving for hundreds of thousands of years, allowing us to adapt to our changing environment. Without evolution, we wouldn't have been able to survive in our developing world. This is responsible for the amazing levels of diversity we see in everyday life. It is a slow process that has allowed us to become the complex creatures that we are today. Evolution is unfolding right before our very eyes, but it is also happening in places within our body that cannot actually be seen. Not only have we evolved physically, but our immune system has had to evolve in order for us to survive attacks from ever-evolving viruses, bacteria, and other pathogens. Our immune system is composed of different antibodies that fight infection from pathogens. Immunoglobulin A is the second most abundant antibody that is found at most mucosal surfaces. These mucosal surfaces include the stomach lining and the rest of the digestive system. IgA can be found in one of two different forms: a monomeric single Y-shaped structure or a dimeric double Y-shaped structure. However, IgA in the dimeric form is more important for the purpose of this video. IgA has an extremely specialised mechanism that is essential to prevent pathogens causing disease. The pathogen penetrates the mucosal layer and enters the organ under attack. Once the pathogen is in the organ, IgA then recognises it as being harmful. To prevent infection, IgA attaches to the pathogen. Once the pathogen has bound to the antibody, the structure can then connect to a receptor that is found on the mucosal layer. This receptor is extremely important in the process of removing the pathogen out of the affected area. Pathogens have evolved in order to strengthen their defence strategies against IgA. Pathogens have been known to secrete proteins called IgA binding proteins. These proteins bind to the receptors of IgA and prevent them from acting upon pathogens. Proteases are enzymes that act like scissors to cut the hinge region from the IgA structure. The hinge region is essential for IgA binding to the receptors on the mucosal surface that are involved in helping eliminate such pathogens. These mechanisms renders the IgA unrecognisable and therefore unable to perform its function. The viruses and bacteria in our bodies are constantly finding ways to evolve and overcome the protection given by IgA at the mucosal surfaces in the body. Once a pathogen evolves, it develops an advantage over IgA. The IgA needs to counteract this process and in turn adaptively evolve to ensure that it can compete with the changes happening in pathogens and fight to stop infections within the body. This can be described as an evolutionary arms race. Both the IgA and the bacterial pathogens that they associate with are in a sort of competition with one another. This means that they are constantly evolving and attempting to gain an advantage over the way the other works. Here is the monomeric structure of IgA. Two of these Y-shaped structures join together to create the dimeric form. Positively selected codons are the areas on IgA that are most prone to evolution. IgA's positively selected codons are highlighted in green. The evolutionary arms race is a constant battle. Pathogens are capable of evolving very quickly, and IgA must keep up. For this reason, the evolutionary arms race will always exist. For evolution to continue, neither one of them should ever win the race.